All right, so on last class, we started talking about cellular division and the process of mitosis. We established that we needed to, our bodies needed mechanisms for creating new cells because our cells, they don't have a infinite lifespan. There's a finite lifespan and we have to replenish our cells. And so we established this fact that we are producing new cells from pre-existing cells. And we highlighted the importance of these new cells being containing the correct genetic material that is transmitted from the parent cell, right? So we talked about the process of cell division and this process of mitosis, right? Which is how we generate new somatic cells. Right. And we said that mitosis basically is a series of steps in which parent cells transmit genetic information to daughter cells. Right. We established that these new cells that are being created are going to have the exact copy and type of chromosomes that the parent cell has. Right. And we discussed what these chromosomes are. We said that these chromosomes contain genes, right? And these genes are basically sequences of DNA that determine specific traits or characteristics about an individual, right? So we have thousands of different genes in our bodies that are spread across our 23 pairs of chromosomes, right? And so we looked at the eukaryotic cell cycle as a means for generating new cells. We highlighted these two main phases of the cell cycle as interphase, right? The interphase is the part of the cell cycle where the cell spends most of its time in interphase, right? And this is kind of that preparation period where the cell is growing and undergoing metabolic activity. It's duplicating its genetic material, DNA replication in S phase. And then G2 is another gap phase where we're just kind of doing some final checkpoints to make sure that everything has happened the way that it should be before the cell proceeds into M phase. Right. And then M phase was characterized by another series of steps where these chromosomes are sort of being manipulated to be dispersed into opposite ends of the cell. And then the actual process of cytokinesis leads to cytoplasmic division. And that cell basically splits down the middle and becomes two new cells. Right. So this is what's happening during the eukaryotic cell cycle. All right, we ran down each of the phases in detail. We're not going to spend a whole lot of time recapping that today, but we want you to remember that what's happening in the eukaryotic cell cycle is that this cell goes through a preparatory phase, which is interphase, and then it goes through M phase where mitosis and cytokinesis takes place. Okay, and so the point here is that the end result of mitosis is one that you should be familiar with, right? The events of mitosis, PMAT, right? We talked about prophase, metaphase, anaphase, telophase, and then cytokinesis, actually leading to two new cells being created, right? So real quick, if you weren't here on Thursday, the take-home message was that after mitosis is complete, all right, mitosis is a part of the cell cycle where we're generating two genetically identical cells, right? The two cells that are created that we end up with at the end of mitosis are going to be genetically identical to the parent cell that it started with, all right? And so we're saying that each daughter cell that is generated from mitosis is going to receive the exact same number and kinds of chromosomes that the parent cell had. Excuse me. Excuse me. Right. And so today, so that's mitosis. Today, we're going to shift our focus to meiosis. Write this down. Meiosis is a special cell division that is producing the cells that are needed for sexual reproduction. All right. So we have a couple objectives for today's discussion. We want to be able to differentiate between these types of reproduction, between asexual and sexual reproduction. We want to be able to distinguish between a haploid and a diploid cell. right? We want to define the term homologous chromosome. We want to be able to explain the significance of meiosis and describe what's happening during this process. And lastly, we want you to be able to now sort of contrast what's happening between mitosis and meiosis, emphasizing the differences in the outcomes, right? So these are two distinct processes, two different purposes, two different outcomes, all right? So let's walk through our reproduction process, all right? So in general, there are two basic types of reproduction in eukaryotes, right? Asexual reproduction and sexual reproduction. You should be able to readily identify the differences between these. Asexual reproduction is when a single parent is involved. It only requires a single parent to generate a new organism, right? So for example, a single parent can split off. It can go through budding or fragmentation to produce two or more new individuals. You see that in organisms like yeast, right? And so asexual reproduction is going to be due to that mitotic division that we kind of discussed before. And so what's happening here is that these new individuals that are being generated inherit the same genes and traits that are identical to that of the parent, all right? This process occurs quite rapidly. It's a very efficient process, as this organism doesn't necessarily need to mate and go through all of the other steps that would be required in a sexual reproduction, all right? And so as we'll see as we move through this talk today, there are advantages and disadvantages to both of these methods of reproduction, all right? So asexual reproduction, we're saying we only require one parent. There isn't any mating going on. And what we're doing is we're duplicating our genetic material and new individuals are being created. These new individuals that are being created are going to be genetically identical to the parent cell that started. On the other hand, we have sexual reproduction where two individuals are required, okay? In the case of humans, sexual reproduction requires the union of two cells, right, that we call gametes, right? Write that term gamete down. Gametes are our sex cells, right? And these are the types of cells that are generated during meiosis. All right. So we said on last class that generally speaking, we have somatic cells in our bodies, right? But we also have sex cells. These sex cells are generated during meiosis. All right. So sexual reproduction requires the union of two cells. And so two different parents are going to contribute these gametes to create a new organism. All right. So for humans, we have mom and then we have dad. So we have the egg cell and we have a sperm cell. All right. So these gametes are going to unite to form a single fertilized cell known as a zygote. Write that term down, zygote. Gametes form two gametes unite to form a zygote. All right. Any questions? And so these two basic types of reproduction, asexual and sexual reproduction, are how we generate new organisms. All right. The results, again, asexual reproduction is going to produce genetically identical organisms, sometimes referred to as a clone. Right. So you can think about asexual reproduction kind of as cloning, right? Whereas sexual reproduction, the results are going to result in genetic variation in the offspring. So every new individual that's created, right, from the union of these sperm and egg cells, they're going to be genetically different. Okay. So offsprings that are produced, write this down, offsprings that are generated from sexual reproduction will not be genetically identical to the parent. Right? What we're going to end up with is what we'll call a hybrid organism, right? Because this newly generated organism or individual, like for the case of humans, we are hybrids of both mom and dad. We get a set of chromosomes, a set of genetic material from mom and a set of genes from dad. So we are not genetically identical to them. We're going to end up being a mixture of genes from both mom and dad. All right, so this is our visual here. I really like to try and find visuals to drive home what we're talking about. So sexual reproduction, we're clear. We start with two parents. Two parents are required for sexual reproduction, and the offspring that is generated will be a mixture of both parents. Okay, that's the result of sexual reproduction. In contrast, asexual reproduction requires only a single parent, right? And the offspring that is created will be identical to the parent cell or the parent organism. Any questions about that? Okay. So real quick, jot these points of comparison down for sexual and asexual reproduction. It would be very beneficial for you to you maybe snap a picture of this, but I'm recording this as well, so you'll be able to come back to it. Sexual reproduction, we know that it requires two parents, right? Two parent organisms, right? In the human, we're going to have the sperm and egg being fertilized to form a zygote. The offsprings that are generated from sexual reproduction are going to be genetically unique from the parents, right? But contain a random combination of parental genes, right? So this is all happening due to random. There's no rhyme or rhythm to which genes you'll get from mom and which genes you'll get from dad. That's all happening randomly, and we'll see that process in just a moment. We talk about crossing over. Sexual reproduction is used by humans and other plants and animals. The advantages, the pros to sexual reproduction is that it's producing genetically different organisms, right? And so anytime you're thinking about genetic variation, that's an advantage, right? That's advantageous when we're thinking about increasing your chances of survival, right? We talked about evolution and these changes that happen over time. Well, genetic variation increases your chances of survival, right? As we think about evolution and adaptation to our environment, being genetically different, having genetic variation in each generation provides survival advantages. One of the disadvantages, though, of sexual reproduction is that this is a very cumbersome process. It takes a very long time. It takes a lot of effort, right? You've got mating happening. You got once this new organism is generated, for example, the human, it takes a whole lot of time for this new organism to be completely formed and developed, right? So it requires a lot of time, a lot of effort in terms of sexual reproduction, all right? Asexual reproduction, we said that it only requires one parent organism, and we're going to have a single organism basically make a copy of itself and then divide. All right. And so the offsprings that are generated are going to be genetically identical to the parent. So whatever genotype the parent has, that's what we're going to see in this offspring. All right. This process is used by yeast, fungi, bacteria. All right. These are all organisms that can utilize asexual reproduction. So the budding, fragmentation, those are used by yeast and so forth. Bacteria utilize binary fission. All right. So advantages, the pros to asexual reproduction. Yes, very extremely efficient process. It happens very rapidly, happens very efficiently, usually without too many problems, right? With great fidelity. However, the disadvantage to that, right, with every good thing, there's a potentially bad thing. One of the downsides to asexual reproduction is that if there are any genetic defects or deficiencies or abnormalities, from a genetic standpoint in that parental organism, guess what? Those same defects and deficiencies will be passed down to the newly created offsprings, right? Because we said that the newly created offsprings from asexual reproduction are an exact copy of the parent cell, right? It's like cloning. So whatever genetic material is there, whatever mutations, whatever deficiencies are there, will be passed down to the offspring, right? So that is an important note to point to note. All right, some of the advantages and disadvantages to these types of reproduction. What questions do we have? Okay, so homologous chromosomes. So we talked about chromosomes, right? We know that our genes are dispersed across 23 pairs of chromosomes. So our chromosomes are going to exist in pairs, right? So in these two partners, if you will, are known as homologous chromosomes. All right. So these homologous chromosomes, these pairs are typically going to be similar in size, shape, and position of their centromere. So I showed you some pictures earlier of what the structure of the chromosome looked like, where we have these two sister chromatids connected by that centromere in the middle. And so these homologous chromosomes are going to carry information about the same genetic traits, right? Those genes that are in our chromosomes are coding for traits, physical traits about us. Information may not necessarily be identical, but the information will be for the same genetic traits, right? So we have these homologous chromosomes. And so this is a structure of, this is what the chromosome structure looks like here. And homologous chromosomes, again, are going to contain DNA that code for the same genes. All right. Homologous regions code for the same gene, although the information may not be identical. So for example, I like to use the eye color analogy because we can easily identify with eye color, but you know, we can all have different eye color, right? Even siblings might have different eye color. That's because these homologous chromosomes they're going to code for the same trait, but the information may not be the same. All right, and so we'll talk about alleles in our next unit when we get to hereditary and genetics. So anyway, again, when we're looking at the cell, we're looking at the genetic makeup of the cell. We have our chromosomes, right? And so depending on the cell type in our bodies, right, we have somatic cells and then we have gametes. Somatic cells, if the cell has two sets of chromosomes, two copies of the chromosomes, it is said to be diploid. Okay, jot that term down. Diploid means that this cell is going to have two copies, two sets of the chromosomes. All right. In humans, that diploid chromosome number is 46. If a cell has two copies of the chromosome, two sets, that cell is said to have a diploid number of chromosomes. The diploid number for humans is 46. That's what we're going to find in our somatic cells. If the cell only has one set of chromosomes, right, that cell is said to have a haploid number. Okay, the haploid number in humans is 23. So I'll ask the question, what cell type in our bodies have a haploid number? I'm waiting for an answer. If our somatic cells have a diploid number, right, 46 chromosomes, which cell types in our bodies only have half of that number, haploid, 23 chromosomes?
Sex cells.
Sex cells, thank you. That makes sense, right? Because our sex cells are required for reproduction. During that process of reproduction, these sex cells must unite with the sex cell of the other parent, right? The sperm must unite with the egg. They both have a haploid chromosome number. Thank you. So for those of us that are visual, a diploid cell will have 2X or 2N, two times, two sets of chromosomes, okay? Two copies of each chromosome, whereas the haploid has only one. All right, so in humans, reproduction, when the sperm and egg fuse during fertilization, each gamete is haploid. It has a haploid number. Each cell is going to contribute one set of chromosomes, right? Which leads to the restoration of that diploid number in that newly fertilized zygote. Okay, so 23 from sperm, 23 from the egg. We now have the restoration of that diploid number. We've restored the diploid number to 46 in this newly fertilized egg. Okay, now this newly formed zygote will continue to develop, right, through and undergoing mitotic division. All right. And these, yes, so I already said that somatic cells are diploid. All right. And so what we're seeing here again is that the diploid number, right, will be 2 times the number of chromosomes that are present for that organism or individual, whereas the haploid number is just N. Okay. Diploid means we're having 2 copies of each type of chromosome that's present in this organism or individual, whereas haploid, we have 1 copy of each chromosome. Make sure you have that in your notes. Okay. So meiosis, let's look at how we actually get these gametes being made. Meiosis literally means to make smaller. Jot that down. To make smaller. Okay. And so meiosis is referred to as a reductive cell division because this cell division reduces the chromosome number by 1/2, right? So we start with these diploid cells, and they're going to undergo 2 rounds of cell division that end up making 4 haploid daughter cells, right? So this is incomplete. This is completely different than what's happening with mitosis, right? We saw mitosis where we undergo 1 round of cell division and we make 2 daughter cells that are genetically identical. Meiosis is a little bit different because first it's going to undergo 2 rounds of cell division, right? And then it's going to yield 4 haploid daughter cells that are going to be genetically different, okay? So not only are they haploid in number, they will not have the same genetic makeup, all right? So during meiosis, there is a process known as crossing over taking place, okay? And so what's happening during crossing over, jot this down, is that these chromosomes are separating and dispersing randomly to these daughter cells that are being generated. All right. This is something we have absolutely no control over. All right. And so crossing over takes place. This is where we're getting our genetic variation. And then we have these 2 rounds of cell division. All right. They are called meiosis I and then meiosis II. Okay. So meiosis I looks just like what's happening during mitosis. Okay. 2 haploid cells are going to be formed from a diploid cell. Okay. Then meiosis II takes place where again, there's going to be separation of these chromosomes into sister chromatids. So really important, the events of meiosis. Let's look at some very important differences, right? Because the goal here is that we're still generating new cells, right? We're undergoing cell division, we're generating some new cells. There are a few differences between mitosis and meiosis, okay? So it's very similar to mitosis, and I see that typo, but there are 4 important differences. The first difference here that I've already mentioned is that meiosis involves 2 rounds of cell division. Okay, those 2 rounds of cell division are going to yield 4 daughter cells. Okay, now, while there are 2 rounds of cell division happening, there's only going to be duplication of the DNA once. Okay, so what we saw in the cell cycle before, you know, G1, SG2, that's only going to happen once. We're only going to duplicate that DNA once, but it's going to be divided twice. Okay. The next difference is each of the 4 cells produced by meiosis are going to contain a haploid chromosome number. Okay, jot that down. Meiosis is going to generate 4 cells that contain a haploid chromosome number that is distinctly different than what's happening during mitosis, right? And then each haploid cell that is generated will have a different combination of genes. They're all going to be unique combination of genes from mom and dad. Okay. So these are 4 very important differences between mitosis and meiosis. Give you a moment to jot those down. Okay. So this is just a visual that shows us what's happening between mitosis and meiosis. We start mitosis with a single diploid cell, right? At the end of mitosis, we're going to end up with two new cells, okay, that are going to have a diploid chromosome number. One round of cell division yielding these two genetically identical daughter cells, which just means that they're going to have the same chromosomal makeup, both cells. Going to be copies of what we started with. Meiosis, on the other hand, is not only going to undergo that first round of cell division, right? But it's going to undergo a second round of cell division, all right? And we're going to end up with 4 haploid daughter cells being created from meiosis. Okay, so we start with the diploid cell. You know, all the processes in the cell cycle are going to take place, interphase into M phase. We're going to end up with our 2 diploid cells being created here. And then another round of cell division will lead to the production of 4 haploid daughter cells. Questions? Okay. So why do we need meiosis, right? The sole purpose of meiosis is sexual reproduction. Okay. Meiosis is required to produce the egg and sperm cells that are needed for sexual reproduction. So the sole purpose of meiosis is to produce gametes. I will ask you about that on your exam next week. All right, so the end goal here is to produce these new daughter cells with half as many chromosomes as the starting cell, right? These daughter cells that we're going to generate are going to be genetically different than each other. All right. So let's look at these, what's going on in meiosis, right? It's a 2-step division process. Meiosis I, Meiosis II, all right? In Meiosis I, we get those homologous pairs to separate. Meiosis II, we see these sister chromatids now separate. We're gonna end up again with these 4 haploid daughter cells with 23 sister chromatids ready for reproduction. Meiosis I, as I said before, Meiosis I looks just like mitosis, right? Also made the point that prior to meiosis starting, the cell must also go through interphase, just like we see in mitosis, right? So interphase happens, we duplicate our DNA, right? We can't skip that step. And then we have PMAT again, all right? So Meiosis I looks just like mitosis. Prophase, we got chromosomes condensing and pairing up. They're becoming visible. Metaphase, we're seeing these homologous pairs of chromosomes line up along the midplate or the center of the cell, right? During anaphase, these paired chromosomes start to separate and move towards opposite ends of the poles. And we know that that's necessary because this cell is getting ready to split off, right? So we saw that elongation of the cell and we see these chromosomes start to separate and move apart. Okay, and so during this,
Do you have anything to say, Malcolm?
I'm sorry? Can you mute your mic for me? During anaphase, the cells are moving apart, and then what's happening is that there's sort of this random distribution of chromosomes now, all right, to opposite poles. And then we had telophase, where we started to see these nuclear envelopes start to form around these new sets of chromosomes. And then cytokinesis occurs, right? So we split off and now we have 2 new cells. That's what's happening during meiosis I. All right. Then meiosis II, it sort of repeats itself, right? PMAT happens again. Chromosomes condense, they align, they move, the chromosomes start to move towards opposite ends of the poles. Nuclear envelopes form around each pole. Cytokinesis occurs again. Now we have our 4 haploid daughter cells at the end of meiosis II. Okay. So what are we getting from meiosis? It's a little bit different than mitosis. Mitosis is kind of like cloning. Everything is going to end up the same. What we get from meiosis is genetic diversity. This is absolutely important. All right. So during meiosis, the exchange of genetic material between these homologous chromosomes is happening. All right. Then we get this random alignment of maternal and paternal chromosomes. So you're getting this random assortment of chromosomes and genes from both mom and dad, right? There's no control over this disbursement. All right. Right. So I want to stop here. These concept review questions I want you to work on for homework. Let me give you a minute to jot these down. You're not going to turn them in, but you need to do them in your notebook for homework. On Thursday, we'll wrap up and have a review session. What questions do you all have for me?